Hello, my name is Christopher Bravery. I'm a consulting regulatory scientist at Advanced Biologicals. And the topic of this talk is to ask the question, are the MSC criteria hindering clinical development? I need to start with a disclaimer. I'm using information that is publicly available from an FDA advisory meeting relating to the BLA for remestemcel-L, which is an allogeneic MSC product for graft versus host disease. I have no current or past involvement with any of the developers of this product. I have no other conflicts of interest, and my intention in this talk is merely to look at the information available and see what regulatory science lessons can be learned from this. And I certainly don't intend any criticism in any of the slides that I put up. So in the interest of the time that I have available for this talk, I'm just going to pick out a couple of these key history moments. So the development of this product, remestemcel-L, started around 2003. After completing 2 phase 3 trials in May 2009, it was concluded that those trials failed to meet their primary endpoints, and so they hadn't demonstrated efficacy in graft versus host disease. The product changed hands. Now, the briefing materials actually say 2011 and 2013, so I'm guessing somewhere in between those two numbers. And it's clear that the company say that they made some manufacturing improvements, and also that they transferred the process from the US to Singapore. They started a new pivotal study, and that study met its primary endpoints, and so they submitted a BLA to the FDA in May 2019. Much of this is based on the briefing materials and the transcripts from the Oncolytic Drugs Advisory Panel meeting, which occurred in August of 2020. So just to set the scene a little bit, in 2014, the FDA published a paper which described the use of the ISCT criteria in 66 INDs, covering mesenchymal cells from various tissue sources, autologous and allogeneic, and over a fair number of years, I don't recall how many. They concluded that on average, developers actually explored 12 plus or minus 7.5 markers for characterization. That sounds a little high compared to my own experience. For release, they were using on average 3 plus or 2 of the markers. And I provided a sort of analysis, I've sort of summarized their analysis or reworked their paper a little bit in this table and included the ISCT criteria, but I don't have time to actually go through that here. So let's remind ourselves what we're trying to do when we develop a product is, first of all, we start with what we think are quality attributes, that is a molecular or product characteristic that is selected for its ability to indicate quality. And then during development, your objective is to try and identify the critical quality attributes. And those are physical, chemical, biological, or microbiological properties or characteristics that should be within an appropriate limit or range or distribution in order to ensure the desired product quality. So while the definition, and that's the ICH definition and the one that the FDA showed at the meeting, well, the definition doesn't implicitly state that these quality attributes should be related to the mechanism of action of the product. By definition, if a quality attribute has to be within a tight range in order to ensure quality, that means it's also to ensure safety and efficacy, which rather implies it should be related to the mechanism of action. And perhaps it should be stated that way, unless somebody can think of an argument why it might not be part of the mechanism of action and yet still meet that definition. So the FDA provided this summary slide, which gives you an idea of the extent of the characterization undertaken by the developers. And as you can see, it does include all the ISCT criteria. And it also includes 2 potency assays. Now, I don't have time to discuss potency. That would be a different talk. But what I should say is, of course, that both of the potency assays, the relevance was questioned by the FDA. So they had concerns about the potency assays as well. But here I want to focus on their concerns around the other critical quality attributes for identity. And their concern that these didn't identify the heterogeneity in the product, or indeed were not identifying the subpopulation of MSCs that has the clinical effect. Now, it says in the briefing book that these attributes have been studied through extended characterization studies of the cell bank, and this is an allogeneic product, so they will have multiple donors, and the drug product, and it's been used throughout development. So while it doesn't specifically say that these attributes remained, these phenotypic attributes remain within any sort of particular range of specification, it's quite clear that there wasn't significant deviations, or they would likely have been commented on, but I can't be sure of that. Now, this is a summary of the release testing, although I've blocked off some of the release testing with the box at the bottom expressing the FDA's concerns. But as you can see, as part of release, they identify and presumably evaluate the purity of the product through CD166 and 105. So CD166 isn't part of the original 2006 Dominici definition of MSCs from bone marrow, which these cells are from bone marrow, but CD105 is. They also just simplify the non-MSC cell types, the cellular impurities, with CD45. But I'm focusing on the phenotypic markers of the cellular active substance here. So primarily they're using just 2 phenotypic markers to confirm the quality of the product. But of course, we saw the other markers that can be used for extended characterization. So this is a sort of an overview of the manufacturing history. And as you can, and all I know is somewhere in this point here where I indicate there were some manufacturing changes. One of them was the tech transfer to Singapore. But it also mentions that process improvements were undertaken. And those improvements led to an improvement in potency, so greater potency. Now, one of the issues with any comparability study is potency should neither be greater nor less. So this in itself, I think, is raising comparability questions in the mind of the FDA, but I'm also not going to dwell too much on that, although I will mention it again. As you can see, the phenotypic markers were the same ones used throughout development, but they have progressed with their evaluation of biological function. So the FDA had concerns about the ability of these quality attributes to demonstrate batch-to-batch consistency. The applicant considers that they have demonstrated consistency by using these markers, so there's a dispute about the relevance of the markers. They also say that the fact that they've used them across development is reassuring because it shows that they have consistency. As I say, there's no mention of them changing, so I assume they have remained the same, and yet the potency increased. So that in itself hints that they are not picking up some of the critical quality attributes of the product. They may be critical quality attributes in themselves, but they're not critical quality attributes that were adequate enough to identify a difference in potency between 2 batches. The FDA also mentioned this aspect of the process changes, and that they had commented that the same critical quality attributes submitted now for BLA had not been accepted during the IND phase. So it seems that the developer didn't really take this on board and just continued. So now let's look in the second half of this talk at just 3 of the phenotypic markers and see what evidence I could find that they might be involved in the mechanism of action of an MSC in graft versus host disease. Now, the first thing I should say here is if you type MSC into PubMed, you get about more than 25,000 publications. And if I add onto that graft versus host disease, I reduce it only to 1,800 publications. And since the ISCT criteria are ubiquitously used for identity of MSCs, it's quite tricky to try and pick out papers that might be looking at the function of these markers in mesenchymal stromal cells, because time and time again you would open the paper and actually that marker was really only mentioned once, just to confirm that the the authors were looking at MSCs. Anyway, so let's look at CD105, endoglin. It's a co-receptor of the TGF-beta receptor complex. It's not required for TGF-beta receptor, but if it's co-localized, it modulates the signal in some way, which gets complicated. A bit too complicated to get to the bottom of entirely. One of the key cells that it's expressed on are endothelial cells. And there is a hereditary disease, which I'm not going to attempt to pronounce, called HHT. And about 90% of patients with HHT, it is because of mistakes in the gene for CD105. And that disease is characterized by multisystemic vascular dysplasia and recurrent haemorrhage, which doesn't entirely suggest it would have a role in GVHD, but of course this would have been something that affected development, so it doesn't mean it's not involved. The TGF-beta family in general, that has an awful lot of genes and different cytokines and so on, and in general terms, the role of TGF-beta is in regulating growth, differentiation, motility, tissue modeling, wound repair, and programmed cell death. So what did I find in the way of literature? Obviously, TGF-beta is added to demonstrate chondrogenesis in the trilineage test. Obviously, I've had a paper or two that demonstrated that. But obviously, that's not particularly relevant or not relevant at all to GVHD. I then found another paper that actually isolated specifically from umbilical cord and adipose tissue MSCs that either expressed CD105 or did not express 105. Now, I didn't spend a lot of time trying to convince myself whether they were still MSCs just because they didn't have CD105, but they did demonstrate that they both produced the same qualitative outcome in the trilineage assay, meaning that the CD105 expression certainly wasn't necessary for the chondrogenesis. And as I said, CD105 is only a sort of a co-receptor along with the TGF-beta receptor, which modulates the signal. So it's not actually required for TGF-beta receptor to function. Indeed, it's not always there. More interestingly, CD105 negative cells actually had a stronger immunomodulatory effect. One paper, though, not corroborated, I didn't find a second paper. Now let's look at CD166 because that's another of their primary markers for release. It's an IG superfamily member, an adhesion molecule. It's known to be a ligand for CD6, which is found on mature T cells, but it is also found on many cells, and I'm not going to read the list out. It's also found in various isoforms, and the full-length one is involved in cell adhesion. And isoform 2, which is missing one exon, is involved in cell dispersion. So this does sort of suggest, well, it's plausible that in MSCs it could be involved with the migration of the MSCs towards the bone marrow for where the transplant is, I don't know, whatever, wherever the cells are that need immunomodulating. But I didn't really find any specific literature of its function in mesenchymal stromal cells. So is it a critical quality attribute for GVHD? All I conclude is maybe. It's not, you can't entirely say it isn't from the little that I could find, but we don't have any positive corroboration that it has a role. So then I looked at CD90. So CD90 isn't routinely used for remestemcel-L, but it is one of the ISCT's canonical positive markers. Its other name is Thi1, which, as you can guess from the name, it's expressed on thymocytes and hematopoietic cells and also fibroblasts. It's another one of these molecules where we're not sure we fully elucidated all its functions, but it is involved in the T cell synapse. If you're aware of that, that comes together to help bind an antigen-presenting cell with a T cell. Does that mean that that's its involvement in MSCs? I guess if T cells are directly engaging mesenchymal cells, that might be the case, but I'm not entirely sure if that's what happens. Is cell-cell contact required? I didn't try and dig into that question, but I think there's a possible role there. If you look at knockout mice, then knockout of Thi1 leads to neurological development problems, cutaneous immune responses, renal development. But again, that is a developmental pathway, and its role in after development can be different. Then I found one paper saying that it is downregulated during apidogenesis, but that doesn't mean to say it wasn't required to start apidogenesis. But what about GVHD specifically? Well, again, I just found one paper. Maybe I could have found more if I'd spent weeks and weeks searching, but I found one paper. And in this paper, they used a hairpin RNA in order to try and suppress the expression of CD90. This wasn't completely effective, so they also did some magnetic bead isolation to get purely CD90 negative, then what I might call CD dim, and it's not what they call them in the paper, and then CD positive. Now, what they noted was that all three populations had a similar level of immune suppression of CD8 cells. Not sure why they only looked at CD8, not CD4, or just a broad group of CD3, T cells. They did notice an increased osteogenic matrix and increased adipogenesis, but chondrogenesis wasn't mentioned, so I'm not sure why. So overall, those results would need to be corroborated by others, but it didn't really look like there was an obvious role in graft versus host. So just to reiterate and to summarize, the FDA's concerns were whether or not these were critical quality attributes. And that included the potency. So together they were really concerned that pretty much all of the attributes identified were not critical quality attributes. They have also concerns around the comparability exercise, which is not surprising if I walk you through the obvious ramifications of not identifying critical quality attributes. You need them for stability. Indeed, you need those critical quality attributes to be stability indicating. If you haven't identified your critical quality attributes, then you can't identify your critical process parameters. You're supposed to do studies to explore the impact of your process parameters on your critical quality attributes in order to demonstrate which are critical process parameters and that you have the process under control. And so without those, you also cannot validate the process, because obviously you don't know what the critical quality attributes are, so you can't do the extended characterization required for validation. And likewise, you can't undertake comparability studies. So overall, my conclusions are the ISCT criteria were never intended for clinical release. So the fact that they're being used was a misuse of an original standard. Whether they are critical quality attributes for any one product or indication needs to be determined for each product and indication by the developer. So this is a gap in the characterization of developers. There's a paucity of data generally in the literature around the role of these membrane proteins in MSC functions. And I would call this a basic science or even regulatory science gap. And it's a little bit surprising, given how much work is being done on MSCs, that no one's really looking at the criteria. Whether a cell is an MSC or not is not per se relevant to its clinical development, actually. All that's required by the regulator is that you can identify the cell that you've isolated, and that you can identify your cellular active substance, which may or may not have the same characteristics as the cell you isolated, and then demonstrate that the quality attributes that you're using are critical quality attributes for the use of that product, i.e. the indication. So until the critical quality attributes are demonstrated, process control can't be confirmed, the process can't be validated, process changes can't be made post-approval. So I can only conclude that by following the ISCT criteria, I think this is hindering clinical development. And thank you for listening.